Hello students, I welcome you all to this online live YouTube session, and this is rapid revision series, and we are going to see 13 sessions with 13 biological topics relevant to the Neet and how to prepare for Neet and all. Okay, so today we are going to talk about photosynthesis in higher plants. So before that, I'm going to give you some tips on how to prepare for Neet. Okay, so whenever you want to prepare for Neet in biology, okay, so in biology you can see botany and zoology, two different types of subjects. You meant to know that botany and zoology. See, both will be comprise questions. Okay, and there are various types of questions in Neet. Okay, the first you have to be very clearly have to understand the topics. What is exactly the topics relevant to the particular subject? That is first one. Okay, the topics. Next one is structure. Okay, and other one is function. Then function leads to technical terms. Okay, so the technical terms. Next, applications. So finally, history based informations. History. So in biology you can see that botany and zoology comprise questions comes in Neet. Okay, when you talk about Neet, you're not going to write exams in answers page by pages. You're going to pick only the perfect answer, the topmost answer. See, when you want to prepare for Neet, you have to be very clear in there is a 4 options. Okay, in that 4 options you have to pick the rightmost top answer. Okay, the 4 answers, all answers are look like a same in biology, in questions when you talk about the Neet especially. Okay, and you can see that you have to prepare like topics, structure, function, technical and application and history. So each and every topic in biology include topics. So you have to be very clear with what is the topic you are going to study. So the topic which is going to be maybe the plant physiology or human physiology, or the general topics like genetics or biotechnology, whatever it is. What is exactly the topic, you have to be very clear with that particular one. And title itself, biology gives you every information. That's very clear. Title itself, biology. See, when you talk about example, plant physiology. Today we are going to talk about a plant physiological function. Okay, the plant physiological functions will include photosynthesis, respiration, okay, growth, and you can see hormones, which are all the hormones which are responsible for that we're going to see. Okay, and before you choose the topic, you have to be very clear. Plant physiology, what is mean by physiology? That is what I told. Topic is very important. Physiology. See, when you prepare for Neet, basic information is very much essential. You can easily prepare deep informations, detailed informations. You will be நீங்க ஒரு தெளிவா இருக்கீங்க, ஒரு எனக்கு எல்லாமே தெரியும்ன்ற ஒரு டாபிக்ல நீங்க இருந்தீங்கன்னா, but you have to be very clear with the basic informations. So plant physiology la irukra photosynthesis, respiration, growth, all these things I know very well. I can solve all the questions if you do like that. But include that, you have to be very clear what is the basic knowledge of physiology. You have to know what is actually physiology. Physiology means there are 2 different types of physiological process occurs. One in plants, another one in animals. So when you talk about the plants, it's very different because the plants are non-motile living organisms. They cannot move from one place to another place. Okay, but when you talk about animal, they are motile. So within that particular topic itself, you have to be very clear what is the word. Physiology is nothing but, you have to be very clear with the word. That's what I'm trying to say. Physiology is the response. What sort of a response? It's a response which you are giving to the external factors, the respiration, digestion, circulation, okay, the movement, locomotion, everything will be the physiological process. You are giving response to the external environment. Ecosystems giving you something, and that will be utilized by your internal body structures of living organisms. That is physiology. So the next one we have seen, structure. Okay, in biology, structure is very, very essential. Structure is very important. See, if you have the clear view, clear idea about the structure, it will be very helpful for you to illustrate the functions from it. So the structure clear-ஆ இருந்துச்சுன்னு சொன்னீங்கன்னா அதிலிருந்து functions ஈஸியாக நீங்கள் derive பண்ணிட முடியும். But functions நீங்கள் ஈஸியாக derive பண்ணிட்டீங்கன்னா the technical terms will be in your mind and hand and everything. So the technical terms are very important. That's we used to call, when you prepare for Neet, it comes under the list of biological term. It comes under the list of biological term. Okay, so biological term. Biological terms are very important. It leads to function. It leads to function. So the function will give you some uses. Okay, that uses will be utilized by the end organism. Okay, so whenever you want to prepare for Neet, you have to follow all these informations. You have to cover with the topics, you have to cover with the structure and function and technical terms and application history. See, there are different types of many questions are involved in Neet. Okay, memory based questions, analytical types of questions, structural based questions, and theoretical based questions and history based questions and all. So we are going to see the 13 sessions inclusive of all this session also. We are going to see the compressions of comprise of all the informations. I mean the topics, the structural information, functional information, analytical information. Analytical means you clearly you can have the idea what is exactly the analytical types of questions. Analytical types of questions you can get from genetics and biotechnology, from those things. Biotechnology அந்த மாதிரியான topicsல நீங்க பார்க்கலாம் analytical types of questions, the pedigree analysis and some other types of you have to analyze and you have to pick the answer, the topmost right answer. That is the analytical types of question. And structural based questions. See, you have to follow the biological terms that will leads you to the particular question, topic and answer. Is it clear? So we are going to talk about the photosynthesis now. Okay, this is our first session. We are going to talk about the photosynthesis in higher plants. Higher plants. So we have to be very clear with all the words. Photosynthesis. Photo means what? Light and synthesis. Something is creating, something is newly creating by converting the light, and the light energy will be converted into chemical energy. Okay, the chemical energy will be utilized for the physiological process for the living organisms, especially the kind of a process where you can see the photosynthesis only or belongs to autotrophic types of living organisms, that is plants. Okay, so the plants are autotrophic, they can produce their own food. Okay, here you can see the higher plants. What about the higher, what is the word here, higher plants? Higher plants. Higher plants, higher animals, these are all the technical terms. You have to be very clear. They are well developed structure, well developed functions, and you can see that they are evolutionarily, they are well developed. Evolution factor, evolution relationship, they are very well much compared to the ancestors. So that is called as higher animals and higher plants. So photosynthesis, light is absorbed and chemicals has been synthesized, and that chemicals will be utilized for the physiological process. The physiological process or belongs to plants comes like respiration, growth and hormone segregation and reproduction process, as well as the transport plants, those things are all comes under the list of plant physiological process. Today we are going to talk about photosynthesis. So based on how I choose the question means I've given you the idea what exactly the kind of a preparation, topic, structural, functional, memory based questions, analytical based question, structural based questions, and we can see the history based and analytical types of questions also I've given. So first question we are going to talk about, this question was asked in Neet 2017, you can see that, and with reference to the factors affecting the rate of photosynthesis, which of the following statement is incorrect? So there is a 4 statements has been given. The question, you have to be very clear with the question. See, immediately you should not go to and see the options when you pick the answer. When you want to pick the answer, you immediately enna pannakoodadhu options la poidu end options correct neenga paakave koodadhu. That is very wrong. You have to be very clear with what is the question. With reference to the factors, what is the factors here? The photosynthesis. What is the see? I already told you that technical terms are very important. Technical terms are very important. In this question, what is the technical term present here? Photosynthesis. Photosynthesis is the topic, you may know that. But this is the question area where you can see the photosynthesis is the kind of a technical term. Photosynthesis. This is one process take in plants, is it not? Okay, this process gives you clear idea to how the plants are producing glucose molecules out of it, and they are utilizing for their need as elements and requirements of physiological process. But when you talk about the photosynthesis, synthesis means what? Newly created. You have to be very clear with the words what is exactly present. Photosynthesis, and photosynthesis is the process. Okay, whenever the process takes place in biology, in living organisms, you can see there is a factors which influence to reduce the reaction as well as the increase the reaction. So the factors, what is the question here? The factors affecting the rate of photosynthesis, that is very important. Which are all the factors affecting the rate of photosynthesis? What are all the factors? What are all the factors? You should be very clear with the information what exactly the factors. Okay, so the factors may be the physical factors, okay, the chemical factors, the environmental factors. Maybe there is a many different types of factors are there. So the factors which are all involved in affecting the rate of photosynthesis. See, if the photosynthesis get affect, the process of the physiological will get inhibit. All the process will get inhibit because the photosynthesis is only creating the glucose molecule which is required by the plants. See, animals can move from one place to another place. They fulfill their needs by foods, by reproduction process, whatever is required as a biological organism. But plants cannot do that. They are stand still in some place until they die. But the growth process will be very high when compared to the other organisms. They started to, once they're born in the world, the growth is unstoppable. So that is the plants. You have to be very clear with that. The photosynthesis and the factors which get affecting the information. Now we are clearly understand the question, what is exactly the factors which is affecting the photosynthesis. So we know what is photosynthesis and what all the factors is getting there are, the beneficial types of factors are all those there. But here the question is the factors which affecting. That is very important. And here the light, first option is light saturation of for CO2 fixation and occurs at 10% of full sunlight. Okay, and tomato is a greenhouse, a crop which can be grown in CO2 enrichment atmosphere for higher yield. So the C3 plants respond to higher temperature, enhanced photosynthesis, while C4 plants have a much lower temperature optimum. And here you can see the fourth option is increasing atmospheric CO2 concentration up to 0.05% of can enhance the CO2 fixation rate. Here you have to analyze the options. What is the option is relevant to that particular question, which actually the words, the factors affecting the rate of photosynthesis. Here the plants affecting the factors which affecting the photosynthesis can be differentiated. There are, you can see, 3 different types of photosynthesis process. generally you can see the three different types of photosynthesis. The first one is we can classify C3 plants. The second one is C4 plants. The final one is CAM. Okay, CAM means what? Xerophytic plants. Xerophytic. I mean the desert la paathirpeenga romba dry areas like grow agara plants. Okay, for that example we can say that Opentia. We can say that Opentia. Opentia stricta is a kind of a species you can see in the Chennai itself. Okay, Opentia. Tamil la sollana chapati kallin solluvaanga. So the Opentia is a kind of a CAM plant which do the photosynthetic process. So three different types of photosynthetic process are there. C3 and C4 and CAM. So what is the difference between C3 and C4 CAMs? Here when the light is getting trapped by the system, I mean what type of system? Photosystem. Okay, there are two different types of system are there, photosystem 1 and photosystem 2. So the photosystem 1 required in some nanometer size of light, and photosystem 2 required some nanometer size of light. That is a quantum physics related information. You have to be clear with that also. So the photosystem 1 and 2, and here the C3, C4 CAM plants. So C3 plants, C4 plants. First you have to be very clear with the information what is C3 and what is C4 plants. That is what we are going to see now. The option 3 is the right answer, because the C3 plants respond to higher temperature with enhanced photosynthesis, while C4 plants have much lower temperature optimum. So the optimum level of temperature will be lower for C4 plants. They are doing the responsible information to that particular plant, C4 plants. What is C3 and C4? It's nothing but carbon 3 molecules getting fixed in these plants. Carbon 4 molecules getting fixed in these plants while the photosynthesis takes place. What is the reactions taking over? It's actually the Calvin cycle. Actually the Calvin cycle. Is it clear? So the C3 plants, you can see that example grains, rice and wheat, and mostly you can see 85% of the plants belongs to C3. In India you can see that all the 85% of the plant species in India, and it occurs in what type of cell? Mesophyll cell. So there is a locative is very important in biology. Locative analysis. Okay, if some process take over, if the physiological process take over, if some biological living organisms doing some physiological process like respiration, like digestion, like reproduction is very important. These are all the physiological process. And what is exactly where the location the reactions take over, which system is relevant to that particular process, what is the chemicals requirement for that particular process, what is the structural events that occur, how many different types of events take over in that particular structures. So that is very important. Here the location of the photosynthesis take over in mesophyll cells. You have to be very clear. Then mesophyll cells na enna meaning, you have to clear with the structure. Plant cell oda structure la neenga clear ah irundinga, mesophyll cells la da C3 plants photosynthesis பண்றாங்கன்றது when you see it. And you have to imagine, okay, so mesophyll cells la da plant physiological process la photosynthesis பண்றாங்க. So you have to be very clear with that particular information, the location information, where exactly. See, we are discussing about the temperature optimum, temperature, higher temperature, C3 plants, C4 plants, everything. But all plants are having cell. And the cellukulla and the reaction enga nadakudhu, adudha romba important. And that occurs in mesophyll cell. Is it clear? So the mesophyll cells occurs containing a C3 plants or doing photosynthesis, and photosynthesis enna produce pandranga 3 carbon molecules. The 3 carbon molecules will be produced by these C3 plants. That's why they are called as C3 plants. 85% of the plants belongs to C3 because the photosynthesis is very similar when compared to the C4 plants. All the 80% of the plants are doing C3 plants types of information. And the stomata which open will move the CO2 in and O2 will go out. So intake of CO2 and liberation of O2. So the liberation of O2 will be going out, the respiration process. But C4 plants, difference, what is the difference between the C3 and C4? It occurs in mesophyll cells. After that, it moves to the bundle sheath cells. So the bundle sheath cells வந்து mesophyll cellsக்கு அப்புறமா உள்ள இருக்கிற layers, the cell structureல பார்த்தீங்கன்னா the mesophyll cells will be present in these areas. See, consider this is a plant cell and you are seeing that it's a plant cell wall, okay? And here you can see these are all the structures, you can comprise it. So mesophyll cells, consider this is the mesophyll cell, this region is the entire plant information, but mesophyll cells are here includes of this, and if you go, the mesophyll cells are doing the Calvin cycle to fixation of C4 carbon into the plants. That's why they are called as C4 plants, and it moves to bundle sheath cells. What is exactly? It moves to bundle sheath cells in the region of the plant cell. So that is the thing you have to be very clear, the C3 and C4 plants. Locative information is dermis region and cortex, and you can see the mesophyll cells over here. This is a stoma. The stomal cells, I mean the stomatal cells. The stomatal cells which closed. And here you can see the mesophyll cells are entering with a CO2 is getting incorporated. And that will be followed by the Calvin cycle to produce C3. Carbon 3 molecules, and carbon 3 molecules will be utilized for the plant physiological process. And here you can see glyceraldehyde 3 phosphate is a molecule which result in the Calvin cycle. You already aware of it when you talk about that. And C3 plants do such types of photosynthetic process, and that is what you have to be very clear with that. And here we are going to talk about C4 plants. C4 plants will be utilized where, that's just few minutes ago I showed you the information, and there is a concentration of CO2 present inside of a mesophyll cell. After that, the photosynthesis process taking place. While that, the duration of those information, the bundle sheath cells will, the CO2 will be moved to the bundle sheath cells, and there you can see the fixation of CO2 take over. So that is C4 plants. There is a production of carbon 4 molecules of the molecules will be utilized. And here you can see the 3 different types of photosynthetic process, and C3, C4 and CAM. The CAM means what? Xerophytic plants. CAM means what? Xerophytic plants. So xerophytic plants, you can see that, and here the duration is different. What is different? Duration. So during the night time, you can see that the mesophyll cells are utilizing the carbon 4 molecules, and daytime the Calvin reactions take over. Okay, so the mesophyll cells, CO2 and all these informations will inclusive of C3 and C4 and CAM plants, and Calvin cycle is the common functions taking over, and because its function is called as photosynthesis. Okay, so C3, C4, CAM, all these will be inclusive. You have to get to know all these informations when you are in a position to giving answer to this question. That is very important. So the option is C3 plants to the respond to higher temperature with enhanced photosynthesis, while C4 plants have much lower temperature optimum. C3 plants எப்பவுமே higher temperatureல என்ன பண்ணுவாங்கன்னா photosynthesis enhance பண்ணுவாங்க, but C4 plants have a lower optimum temperature receive பண்ணுவாங்க. So the C3 plants are mostly in India. India is the country where you can see the tropical country. So moreover, like 1 or 2 months only will be very cold and winter and rainy seasons, but other 8 and 9 months are பாத்தீங்கன்னா ரொம்ப ஹீட்டான ஒரு சென்சேஷன் நம்ம ஃபீல் பண்ணுவோம். So tropical country like India, C3 plants are moreover they are doing that higher temperature enhanced photosynthetic process. So that is the first question we have seen today, and we are going to see the next question. What is exactly we are going to see? The water vapor comes out from the plant leaf. You have to imagine we are talking about the plant leaf in stomatal opening. Stomatal open ஆகிறதும் close ஆகிறதும் ரொம்ப முக்கியமான function plantல, because the diffusion of oxygen, the diffusion of CO2 will take over. So the stomatal opening process and carbon dioxide diffuses into the plant during the photosynthesis on the same opening of the stomatal region. So அதாவது இந்த கொஸ்டினில் என்ன சொல்கிறாங்க அப்படின்னு பார்த்தீங்கன்னா, stomatal open ஆகிறதும் ஸ்டொமேட்டல் ஓப்பனாக இருக்கும்போது கார்பன் டை ஆக்சைடு டிஃப்யூஸ் ஆகிறதும் அண்ட் வாட்டர் வேப்பர் கம்ஸ் அவுட், வாட்டர் ட்ராப்லெட்ஸ் அதுவும் எல்லாமே வந்து பார்த்திங்கன்னா ஒரே டைமில் நடக்கிற இன்ஃபர்மேஷன். அதுவும் பார்த்திங்கன்னா இட்ஸ் டேக்ஸ் பிளேஸ் இன் த சேம் பிளேஸ். ஸ்டொமேட்டல் ஓப்பனாக இருக்கும்போது என்னவாகும்? கார்பன் டை ஆக்சைடு டிஃப்யூஸ் ஆகும். during the photosynthesis. And plant leaf la enna agum droplets create agum, that water vapor. What is meant by water vapor when you talk about water vapors? Students, water vapors, transpiration. Transpiration is the process where you can see the excess amount of water molecules, water content present in the plant system will be oozed out to the stomatal region. So the leaf material irukku illingla, the leaf material surface la irukra stomatal open moolama the excess amount of water will be come out. So the water will be come out to take up the transpiration pull process. So the transpiration pull process initiate the root system to suck the water very fast. So that is the thing you have to be very clear in this process. See, within the question we can discuss the entire topic, so that every question is important. And the photosynthesis planned during while diffuses information. And here this is the question which is asked in 2016 Neet. And both process can happen together because the diffusion of coefficient of water and CO2 is different. And next is both process cannot happen simultaneously. And one process occurs during the daytime and other at night. Here you have to be very clear with all the informations. The above process happen only during night time. See, what is exactly the kind of a process here relevant to this particular question? Both process, rendu process irukku. Enna rendu process? Water vapor, transpiration process, and stomatal open ஆகும்போது photosynthesis taking place and carbon dioxide getting diffuses. Carbon dioxide is getting diffuses. What is mean by diffuse? Diffuse na enna? The diffusion means what? The something's getting moved from the higher concentration to lower concentration. So the higher concentration to lower concentration move ஆகுறப்ப என்ன ஆகும்? So அங்க rapid-ஆ ஒரு region-ல molecules will be reduced and another region la, opposite region la, molecule will be increased. So the kind of a process here, the option is both process can happen together because the diffusion of coefficient of water and CO2 is different. See, diffusion of water, coefficient of water and CO2 is different. What does exactly mean? You can see the transpirational loss of water occurs through the stomata and gaseous exchange also occurs through the stomata. ஒரே ஒரு ஸ்ட்ரக்சர் தான், பட் அந்த ஒரு ஸ்ட்ரக்சரில் பார்த்தீங்கன்னா you can see both the functions. Stomatal ஓப்பனாக இருக்கும்போது transpiration of மூலமாக water vapors will be come out and gaseous will be exchanged. Gaseous exchange means what? The CO2 will be intake and O2 will be liberated. So the O2, how exactly the O2 is getting out of the plant system? Hydrolysis process, the water splitting process. H2O will be H2O will be removed in the hydrogen ions and O2 molecules. The hydrogen ions will be utilized for the rest of the reactions takeover in photosynthesis in the thylakoid region of the chloroplast membranes. You already know it. And the O2 will be liberated out of the plant system, and it's simultaneously the process takeover. In the process will be simultaneously takeover with simple diffusion process. The simple diffusion and the meaning higher concentration to lower concentration. வெளியில் இருக்கிற CO2 is getting incorporated into the plant system. The same way stomatal opens is also giving you the way of inner higher concentration to lower concentration. The water molecules will be considered as excess amount of water content will be removed out of the plant system through the stomatal opening. So that is why you can see the different diffusion pressure gradient and diffusion coefficient. See, the pressure gradient means what? ஒரு pressure create ஆகுது. என்ன pressure create ஆகுது? Plant systemல transport in plants process la, root system will be absorb the water content will be come out of the plant system through the stem region, leaf region, branch region. So அந்த region க்கு வந்தது என்ன ஆகும்? ஒரு transpiration pull process create ஆகும். And the transpiration pull process எப்படி, how it gets created? Through the transpiration pull process will be include the water is getting sucked up the reaction. That's nothing but the bulk movement. அது என்னன்னு சொல்லுவாங்கன்னா பல்க் மூமெண்ட் ப்ராசஸ் சொல்லுவாங்க. வாட்டர் மாலிகுல்ஸ் பல்காக என்னவாகும்? மூவ் ஆகி மேலே போயிட்டே இருக்கும். ஸோ இட்ஸ் அப் த ரியாக்ஷன், இட்ஸ் சக் அப் த ரியாக்ஷன் ப்ராசஸ். தட் பர்டிகுலர் வேர்ட் இஸ் ஹைட்ரோஸ்டாட்டிக் ப்ரெஷர். Hydrostatic pressure. Okay, transpirational pull is relevant to that hydrostatic pressure because leavesல இருக்கிற stomatal open இருக்கும்போது excess amount of water will be come out, that is water vapor. Okay, so அந்த water vapor அந்த regionலயே என்ன ஆகுதுன்னா CO2s get diffused. So the water move ஆகிற அந்த process நம்ம என்னன்னு சொல்கிறோம், hydrostatic pressure சொல்கிறோம். ஹைட்ரோஸ்டாட்டிக் ப்ரெஷர்னா மீனிங் உங்களுக்குலாம் நல்லா தெரியும் அந்த விஷயம். ஹைட்ரோஸ்டாட்டிக் ப்ரெஷர்னா நீங்கள் எல்லாருமே டெண்டர் கோகோனட் சாப்பிட்ருப்பீங்க. ஸோ இந்த டெண்டர் கோகோனட் சாப்பிட்றப்ப என்ன பண்ணுவீங்க, ஒரு ஸ்ட்ரா வச்சுட்டு you suck the tender coconut water inside இல்லையா. ஸோ அப்படி பண்ணுறப்ப என்ன ஆகுது, ஒரு ப்ரெஷர் க்ரியேட் ஆகி உள்ளே இருக்கிற கண்டென்ட் டியூப் வழியாக மேலே வந்து நம்ம மவுத்துக்குள்ளே போகும். The same way the pressure gradient is nothing but the hydrostatic pressure is the direction of the molecules, the content, water content will move towards the upper part of the plant. And the upper part of the plant க்கு போகிறதுக்கு யார் யூஸ் பண்ணுறாங்க, transpirational pull process. So the transpirational pull process gives you the clear idea what exactly the hydrostatic pressure. So the hydrostatic pressure leads to all the parts of the plants will receive the enough amount of content, water content. Excess amount of water will be removed. அது remove ஆகி வெளியில் போயிடும் stomatal open இருக்கிறப்போ. The same way the CO2 is also get diffused. Pressure gradient என்ன meaning, what is that? The pressure gradient is the direction of the particular molecule is getting moved out of the system. So that is the pressure gradient. You have to be very clear. Diffusion coefficient, that is the pressure gradient, both the same. So both the process can happen because there is only one structure. You can see the structure very clearly. Here the stomata is getting open here. This stomata open, stomata enna solranga, swollen solranga, and shrunken stomata, it's closed, and stomata closing. These are all the God cells surrounded by that. Here you can see the water. Okay, water and molecules. This is the normal cell of the plant present in the epidermis region of the leaf. So it contains all the normal cell organelles inside it. So don't worry about that. And here you can see the stomata open and stomata closed. So these both things will give you the clear view exactly. This is the place where the CO2 is getting diffused with the pressure gradient point, and the water vapor transpiration pull process also take over. So the option is one, both process can happen because the structure is one but the process is two. Okay, so next question we're going to talk about Oxygenic photosynthesis is characteristics of. Okay, so what are all, you have to be very clear, this is the technical term, this is the biological term, students. Okay, this is the biological term. Photosynthesis, you already well aware of it. You have to break this word oxygenic. Okay, there is a reactions takeover oxygenic. This is normal photosynthesis. This is normal photosynthesis. When you talk about the anaxogenic means what? Different types of photosynthesis respond to the different species that we are going to talk about. Okay, Spirogyra, Chlamydomonas, Ulva, these are all the things you have seen. The bacteria, I mean what sort of bacteria? They are photosynthetic bacteria. Photosynthetic bacteria, and these bacteria are always do photosynthesis because they have pigment. They are also belongs to the autotrophic mode of nutrition, and you have to be very clear with information. Basic information is much essential in Neet. Spirogyra, Chlamydomonas, these are all the organisms or belongs to the algae types of bacteria. Okay, photosynthetic bacteria. And இவங்கெல்லாம் என்னன்னு சொல்கிறாங்க அப்படின்னு பார்த்தீங்கன்னா, ஹையர் பிளான்ஸ் இருக்காங்க இல்லையா, they do photosynthesis, they do reproduction, flowering, everything. These are all the organisms which are called as ancestors of those higher organisms, because the photosynthesis only the only thing which take over in Chlamydomonas and Spirogyra and all those, the photosynthetic bacteria take over. The same process will be take over in higher plants also. So what is exactly these are all the types of information is very dissimilar. It's இங்க சம்பந்தமே இல்லாம ஒரு இன்ஃபர்மேஷன் இருக்கு. அது என்னது? Rhodospirillum. Spirillum means what? Spirally structure arranged bacteria. Okay, so the rhodospirillum means what? Rhodo na enna meaning? The option is purple non-sulfur bacteria, rhodospirillum. Before we are going to that, you have to be very clear with that each Spirogyra is doing what sort of photosynthesis? Chlamydomonas belongs to what sort of photosynthesis? Vulva was exactly. And rhodospirillum, what exactly the kind of bacteria? You have to include, you have to get into all this information is much essential. And here, anexogenic types of photosynthesis. Here, the rhodospirillum is a purple non-sulfur bacteria. So the purple non-sulfur bacteria, the bacteria which contain the chlorophyll, that's contain the chlorophyll is called as bacteriochlorophyll. So the bacteriochlorophyll na enna pathina, they are similar to the chlorophyll. See, the rendu reactions you can see that. Rendu reactions. What is exactly oxygenic photosynthesis and this is anexogenic photosynthesis. This is very normal. You can see the production of glucose from the light energy and water will be utilized for that, and the result of these H2O and oxygen will be produced, and the carbon dioxide is incorporated into the plant system by doing photosynthesis, and oxygenation process take over. And here the light energy will be utilized for that. This is normal. This is very normal. And what is here? See the kind of bacterias, okay? The kind of bacteria need to do photosynthesis. Very important. Listen. The kind of bacteria are do photosynthetic process. So the photosynthetic process, normal plant vandu photosynthesis, how they do, how do they do that? The normal photosynthetic bacteria will be utilized the light energy, okay? Utilize the light energy and they convert it into chemical. They convert it into chemical. Here, electron donor is very important in photosynthesis. Electron donor is very important. Electron donor anada, electron donor accept panada, the energy will be produced. What energy will be produced? ATP and NADPH will be produced. So ATP and NADPH produce anada, anga glucose molecules produce panna mudiyum. The glucose molecules are produced பண்ணாதான் photosynthesis ஓட result அங்க fulfill ஆகுது. So the photosynthesis will be produced by, the photosynthesis process will be producing glucose molecules, and here you can see the anexogenic photosynthesis. என்ன meaning? Direct என்ன ஆகுது? They are electron donor is the organic component இருக்கணும் photosynthesis processல. But autotrophs cannot extract the electrons, negative particles from water, because we are talking about the rhodospirillum, chlamydomonas, vulva. The vulva is different. Sporagora and chlamydomonas, rhodospirillum, these are all the bacterial types of living organisms. And that you can see the rhodospirillum, you can see in water content la irukkrappo enna aagudin pathina, water la irundhu enna panna mudiyadhu, electrons derive panna mudiyadhu. Electrons molecules will not easily derive from the water. Extract பண்றது ரொம்ப கஷ்டம். So the autotroph types of bacteria cannot extract the electrons from the negative particles from the water, because the use of electrons from an inorganic and organic by bacteriochlorophyll. The chlorophyll structure will be different when compared to the normal plants and photosynthetic cyanobacteria. But bacteriochlorophyll is similar to the chlorophyll structure. Here you can see two different types of photosynthetic process. One is anexogenics, another one is oxygenics. Anexogenics na enna pathina, electron donor anexogenic photosynthesis la inorganic or organic materials choose பண்ணி அதில இருந்து என்ன பண்ணிடுவாங்க, electrons extract பண்ணிடுவாங்க. So by that time it will be used. Here you can see hydrogen sulfide is the electron donor. In hydrogen sulfide you can see that, okay? And carbon dioxide will be incorporated, light energy will be utilized, electron donide you can be used it. Here you can see hydrogen sulfide, and these are all the electron donor. These are all the electron donor, and they are reduced inorganic electron donor. That is very important. They reduced it and they can use it. And carbohydrate is also getting produced out of it, and water will be produced. The ultimate aim is carbohydrate when you do for photosynthesis. Here the glucose will be created, carbohydrate will be created. And this is anexogenic photosynthesis. Only occurs in types of organisms where you can see the organisms which cannot extract the electrons from the negative particles of the water. So if they can extract, they can do the normal types of photosynthesis process, that is oxygenic photosynthesis. Okay, is it clear? So the photosynthesis process involving these types, in according to the species to species, it's get differ. So now next we're going to talk about the emulsions enhancement effect, and red drop have been instrumental in the discovery of. So what exactly? The photophosphorylation of non-cyclic electron transport and two photosynthesis operating simultaneously, and photophosphorylation and cyclic electron transport, oxidative phosphorylation. Two photosynthetic processes operate simultaneously. That is the right answer. Why? Here you can see red drop process enna nadakudhu, photosynthesis process nadakudhu. Immersement enhancement effect, that means triple E. That means triple E process. That is also takeover in particular place. Here you can see 700 nanometer size. They are, see the wavelengths beyond 700 nanometer. Wavelength beyond 700 nanometer irundhuduna, nanometer very very minute when compared to the size of, you cannot see that naked eye. And here the photosynthesis process la energy drive பண்றது ரொம்ப ரொம்ப கஷ்டம் when they go for 700 nanometer. So some point that huge drop efficiency take over in 700 nanometer, it's beyond the limit. That's called as red drop effect, because you have to very clear with information. Photosynthesis will be absorbed by the plants, two different types of lights. One is far light, another one is red light, one is far red light. One is red light, another one is far red light. Okay, so two different types of red light will be absorbed by the plants. So beyond the 700 nanometer size la ஒரு process நடக்கும்போது அங்க huge efficiency will get dropped down. That is red drop effect. So that red drop effect is also takeover, and you have to be very clear with the information. 680 nanometer, 700 nanometers are two different types of photosystems, photosystem 1 and photosystem 2. And here the decrease in quantum yield. Quantum yield is important because see the wavelength of the sunlight is important for the light to trap and convert it into chemical energy. That is the important thing you have to be remember. The number of oxygen is also released for light quantums absorbed in the quantum yield of photosynthesis. Photosynthesis normal nadakara process la red drop effect nadakum. At the same time, what is exactly going to happen? Triple E process nadakum. Triple E process na enna meaning, the quantum, the short light, shorter wavelengths alternate patients la move ஆகி வரும். So that is noted by, here you can see the enhancement is known as triple E, immersion enhancement effect. So in the evidence, the first enna பண்ணாங்க, photosystems absorb the first far red light, then red light. So from that it will be concluded red light and far red light, two different types of lights. Okay, and two different types of functions take over, and that will be present in two different types of functions in one process. That is photosynthesis. Okay, so the quantum yields are nothing. They are actually expected rate la irukrangalandradhu problem kadiyadu, but the efficient of two different types of functions take over in one process. That is, you can see the red drop effect as well as the triple E, that's called as immersion enhancement effect. So these two processes will give you the clear view of two photosynthesis operating simultaneously in enhancement effect, immersions, and red dropped information. Is it clear? And now we'll move on to the next one. So during the light stage of photosynthesis, the photoactivated pigment removes an electron. See, when the light is getting trapped by the photopigments, the photopigments oda work enna, light trap ஆன உடனே electrons excite statement க்கு வந்துரும். Electrons excite statement க்கு வந்துச்சுனா it's move from one place to another place. அந்த மூவ் ஆகிற electrons என்ன ஆகும் அப்படின்னு பார்த்தீங்கன்னா, it go and bind with the electron carrier. So the electron carrier என்ன பண்ணும், ஒரு காம்பனென்ட்ல இருந்து இன்னொரு காம்பனென்ட்க்கு எலக்ட்ரான்ஸை கொண்டு போகும். இந்த கொண்டு போகிற இந்த பாத்வேல ஏடிபி ப்ரொடியூஸ் ஆகும், NADPH ப்ரொடியூஸ் ஆகும். தட் இஸ் த photosynthesis process. And here you can see the fate of free hydroxyl radical is that it is broken down into oxygen and free radical of hydrogen, and raise the activation of level of chlorophyll by donating the positive charge, or is to produce adenosine triphosphate from adenosine diphosphate, or reduced carbon dioxide to sugar. So option is 3 is the right answer, because during the light stage of photosynthesis, the photoactivated pigments removes an electron from the hydroxylation derived from the water molecule. What is hydroxylation derived from the water molecule? Water molecules will be absorbed by the root system, will come to the top of leaf for photosynthesis. So the water splicing process take over. H plus ion agum. I can clearly give you the information now. Okay, so the water splice process, H2O will be removed in the H plus ion plus O2. So the O2 will be removed out of the plants, so the H plus ions. So here you can see the hydroxylation derived from the water molecule, and hydroxylation derive ஆகுறப்ப என்ன ஆகுது, electrons வந்து pigment remove பண்ணிடுறாங்க. இந்த pigment remove பண்றப்ப என்ன ஆகுதுங்க, ATP will be converted from the adenosine diphosphate. So adenosine diphosphate from adenosine triphosphate will be produced, and that ATP is the produced molecule, energy molecules required by the plant system. So exactly, you have to be very clear with the questions and understanding concepts. And here you can see hydroxylation is a kind of a technical terms you have to remember. Hydroxylation, what is exactly the hydroxylation, you have to be very clear with that. Technical terms therinjale, the technical terms leads to the topic, function, structure, you can easily get the answer. And here we're going to talk about the diagram which clearly shows the ATP synthesized through chemoosmosis. I already told you that basic information is very important, students. Basic on information is நமக்கு ரொம்ப clear-ஆ தெரிஞ்சிருக்கணும். When you talk about, when you want to prepare for exam, not only for Neet, when you want to prepare for exam, basic information தெரியாம நம்ம deep-ஆ தெரிஞ்சு வச்சுக்கிட்டோம்னா it will be no use. So the basic information is you have to be very clear with the questions what exactly is there. Diagram below shows the ATP synthesis and through chemoosmosis. You have to be very clear with what is osmosis, what is chemoosmosis. Osmosis means what, chemoosmosis means what. In the in isotonic solutions, some particles will be moved from a higher concentration to lower. So and chemicals which involve in that particular process. And here the picture what exactly it gives you, chemoosmosis, chemical reactions, chemicals movement from one region to another region. And here you can see the question, A is here, B and C, D. You have to verify what is exactly the process here. The which option shows the correct labeling A, B, C, D in the diagram. This is, you can see the interior of the thylakoid. Thylakoid is the location information. I already told you that location is very important in biological living organism process when you want to study that. And here the ATP is getting synthesis. ATP synthesis is involved in the production of ATP molecule. I already told you that more hydrogen ion molecules will be included in the process of photosynthesis when there is a photosynthesis process take over, because the water splitting process, hydrogen ions can be produced and the O2 will be removed. And here you can see NADPH getting produced here. H plus ions, hydrogenic ions is getting inside of the thylakoid region, and this is stroma region. This is thylakoid membrane. You can see the membrane, thylakoid membrane here, and that's what we are going to see. The thylakoid membrane is here. You can see photosystem 1 and thylakoid membrane and F naught. F naught means what? You have to be very clear with each and every individual word. Why do I am saying this? Photosynthesis process gives you the inclusive of all the complex structure. It's a very minute microscopical and intra level of intracellular functions they are. They are called as intracellular function. So here you can see the photosystem 2 and photosystem 1, and F naught is here and thylakoid membrane. So inside thylakoid membrane you can see something is getting incorporated. That is the word they are mentioning, chemoosmosis. And NADP converted into NADPH by the process of photosystem 1 and photosystem 2 here. Cytochrome BF. What is this actually cytochrome BF? They are catalyzers. What sort of catalyzers? இங்க என்ன reaction நடக்குது catalyze பண்றதுக்கு? Reaction is here. Reaction is converting the ATP, producing the ATP by using what? Photolight. That is photosynthesis, where it takes place inside a chloroplast. And here you can see the ATP synthase will be inclusive of ATP into ADP converted into ATP. And here the F naught is nothing, the fluorescence, the light condition which absorbed by the pigments. Okay, light absorb ஆகிற அந்த ரேஷியோ இருக்கு இல்லைங்களா, எவ்வளோ லைட் உள்ள எவ்வளோ லைட் intensity will be given to the particular structure of a chloroplast. That is will be indicated in the form of F naught. The F naught is nothing but minimum low light எவ்வளோ absorb ஆயிருக்கு. அதுதான் F naught. And the F naught will be included in the thylakoid membrane region, where you can see catalyze the transfer of electrons from a plastoquinone to plastocyanone. Plastocyanone, these are all the electron carriers. They are actually called as cytochromes, electron carriers. The electron carriers function only to carry the electron which excited from the light energy trapped by the photopigment. That's very important. Photopigment என்ன பண்ணும், லைட்டை ட்ராப் பண்ணும். லைட்டை ட்ராப் பண்ணும், எலக்ட்ரான்ஸ் என்ன பண்ணும், எக்ஸைட் ஆகும். எக்ஸைட் ஆகி எலக்ட்ரான்ஸ் மூவ் ஆகும். மூவ் ஆன உடனே cytochrome B and F will be carry the electron, and that electron will be moved to photosystem 1 to 2 and 2 to 1, like that. There you can see that non-cyclic phosphorylation, cyclic phosphorylation, where it completes, where it's not complete. So those things are all comes under here. This question gives you clear idea where the location is. You ought to be very clear with the chloroplast structure inside what is exactly the thylakoid membrane and thylakoid region, how exactly where the location of those processes take over. That is very important. So the thylakoid membrane, photosystem 2 and photosystem 1, option 4 is the right answer. See, direct turning option kulla ninga pogave kodadu. You have to get to understand the question first. What is exactly the structure? Structure என்னென்ன காம்பனன்ட்ஸ் இருக்குது, என்னென்ன ரியாக்ஷன்ஸ் டேக் ஓவர், என்ன டைப்ஸ், எனி டெக்னிக்கல் டேர்ம்ஸ் எதனா இருக்கா? டெக்னிக்கல் டேர்ம்ஸ் இந்த ஸ்ட்ரக்சரில் கிடையாது, பட் என்சைம் நேம் கொடுத்துருக்காங்க. ஏடிபி and synthase. ATP synthase will be used for ATP is converted into ADP is converted into ATP, adenosine triphosphate. இது எதோட ரிசல்ட்டாக இருக்கும்? பிளான்ட் உள்ள photosynthesis process result. And this result will be utilized to create glucose. That is different. Light reaction, dark reaction, two different types of reaction, and that will be interior of a thylakoid. Thylakoid region எங்க இருக்கு? குளோரோபிளாஸ்ட்ல இருக்கு. குளோரோபிளாஸ்ட்ல இருக்குன்னா அப்போ you have to be very clear. குளோரோபிளாஸ்ட்ல இருக்கிற அந்த ரீஜன்ல தான் எக்ஸைட் ஸ்டேட்டட்ல எலக்ட்ரான்ஸ் மூவ் ஆகும். அந்த எலக்ட்ரான்ஸை யார் ட்ராப் பண்ணுவா? electron carriers trap பண்ணுவாங்க. So electron carriers trap பண்ணி மூவ் பண்ணி கொண்டு போறப்போ there is a production of NADPH and ATP molecules will be required for the formation of glucose inside of a plant system. Okay? We are going to see the next question, which is a very simple question, but it's very important. Different types of memory-based types of question. I've already told you that memory-based types of question and history-based types of question, structural questions, analytical types of question, and theoretical and technical term questions. But here you can see the pigment-containing membranes, extensions in some cyanobacteria or basal bodies, pneumatophores, chromatophores and heterocyst. See, basal bodies, pneumatophores, chromatophores and heterocyst, these are all the parts of structure taking over in cyanobacteria. You have to be clear with that. So cyanobacteria are the structures on your mind la irukanum. Exactly what is cyanobacteria, how exactly they look like, what is the pigment present, what is the reproduction, what are all the species name, all those things will be in your mind. Cyanobacteria and heterocyst, chromatophores, pneumatophores, basal bodies, these are all the structures enama irukalam. But here you want to talk about pigment containing membrane. What is it exactly? Pigment containing. Pigment leads to what? The pigment of chlorophyll A, B and carotenoid and xanthophyll. These are all the things. So these are all the pigments information you have to be very clear. And heterocyst, you can see the special types of outgrowth. It's a special types of structure present in the the filamentous types of cyanobacteria. You can see heterocyst types of structures. So that is the most important place where the nitrogen getting fixed in the cell from the atmosphere. So the chromatophores also give you the clear information here, the pigment containing membrane extensions in some cyanobacteria, because it's contain photoautotrophic bacteria possess chlorophyll, and that chlorophyll will give you the extension of information. Chromatophores ninga pathina or consider this is a kind of cyanobacteria which is doing that, and in that session you can see on the cytoplasm region you can see the chromatophores like this. Okay, so the chromatophores will be present, and they are the responsible information to produce the photosynthetic process to start. So that is very important. And basal bodies, pneumatophores and chromatophores and heterocyst. Chromatophores are the important answer, and the topmost answer also. The chromatophores are the reason where the photosynthesis takes place, students. Okay, because the question clearly gives you pigment containing. Why there is a pigment? Pigment means what exactly? Pigment. You have to be very clear with all the words. Technically is very important. Pigment. Pigment nale you have to be 100% proteins. The proteins which are responsible for the structure and function. Pigmented proteins are involved in the photosynthetic process. Is it clear? So the chromatophores are the pigment containing membrane extension in cyanobacteria. Now we're going to talk about the two plants Na and B are supplied with the O2 and H2O18 and CO218 with the H2O respectively, which following the plant releases O18 type of oxygen photosynthesis. In this question you have to be very clear. O18 kuduthirukanga illaya, there is a O18, H2O18 is there, right? H2O18 is there, and here CO218 is there. See, these are all the words which make you confuse. Ungala confuse பண்றதுக்காக கொடுத்திருக்கிற words அது. You should not get confused by seeing these types of in the numerical information. Okay, is it clear? So the 18 and H2O18 process. So which following the plant releases the O18 type of oxygen photosynthesis, and you have to be very clear. Photosynthesis process nadanduchina, what is the process will take over? The H2O will be absorbed by the root system, will be used for what? The photolysis process, water splitting process. H2O enna agum? H2O will be converted into H plus ions and O2 will be out of the plant system. Here the photosynthesis process, here the O2 and H2O18 and CO218, H2O respectively. This is information you have to be very clear. The H2O18, these 18, these are all the words makes you confuse. You have to be very clear. A plant, B plant, first A, then B, and both A, B plants. Here photosynthesis la or information enna H plus ions agum, O2 agum, thaniya enna பண்ணுவாங்க, information carry forward agum. So the O2 is nothing but water splicing reaction. So the water splicing reactions la you can see that enna madriyana numbers of 18 whatever is this. Okay, so O2 will be removed, CO2 will be intake, and with H2O respectively. And H2O irundadhan there is going to be water splicing reactions take over. But when you talk about this question, two plants A, B, so during photosynthesis enna nadakum? O2 will be evolved. That is the normal process, natural process. You can see hydrolysis of water, and if H2O is used in a plant A, then it became true that O18 type oxygen will be evolved from a plant A. So the enna reason anga solranga, whether it's going to be a plant A or plant B, H2O anga use pannirundala enna agum? O2 will be evolved. So that is the function. This is very normal question, normal function, but it's confusing you by giving you the 18, O18 and all will be given to you. So you have to be very clear with that. You should not get confused by these questions. Okay. The next we're going to talk about how many molecules of ATP and NADPH are required to respectively to make 3 molecules of glucose. Okay. See here, ATP, NADPH, these are all energy molecules. In the energy molecules edukku produce agudhu? To create what? Glucose. Glucose yen create panano? The plants are doing photosynthesis process pannada they can get food out of it, because they belong to autotrophic. You have to analyze when you see a question all this information. அப்படி கொஸ்டினை பார்த்த உடனே உங்கள் மைண்டுக்குள்ளே வரணும் இந்த ப்ராசஸ் எல்லாமே. அப்போ தான் நீங்கள் நல்லா ப்ரிப்பேர் பண்ணியிருக்கீங்கன்னு அர்த்தம். The molecules of glucose. The molecules of glucose will be created on the photosynthesis process considered as a food. என்ன எதை யூஸ் பண்ணி கன்சிடர் பண்ணுறாங்க? ATP, NADPH. ATP, NADPH எப்படி ப்ரொடியூஸ் ஆகுது? photosynthesis லைட்டை ட்ராப் பண்ண உடனே எலக்ட்ரான்ஸ் எக்ஸைட் ஆகுது, எலக்ட்ரான்ஸ் கேரியர் மூலமாக ஃபார்வர்ட் ஆகி ATP, NADPH ப்ரொடியூஸ் ஆகுது. ஸோ இங்கே கொஸ்டின் என்ன? How many molecules of ATP, NADPH is required to create 3 molecules of glucose? 3 molecules glucose create ஆகிறதுக்கு எவ்வளோ ATP, NADPH ப்ரொடியூஸ் பண்ணுறாங்க? See, you have to be very clear. There is a, you can say that 3 ATP in NADPH required. 3 ATP molecules 2 NADPH molecules are required. எதுக்கு 1 CO2 molecules are fix பண்றதுக்கு. But here the question is 3 molecules. Question is 3 molecules. 3 molecules of what? Glucose. Here you can see the 3 molecules of glucose will be produced by the 3 molecules of glucose will be produced by the ATP and NADPH. But how many molecules are required? That is question. You have to be very clear with the question. 54, 36, 36, 36, 18, 12, 24. Here you have to be very clear. 3 ATP is required, 2 ATP is required, 2 NADP is required to fix the 1 CO2 molecule and produce glucose. Okay? And here what is exactly the kind of a the glucose molecules or the structures, and what is the glucose molecule structure? C6H12O6. So C6H12O6 தான் glucose molecules. So glucoseல எத்தனை கார்பன் இருக்கு? 6 கார்பன் இருக்கு. ஒரு கார்பன் மாலிகுல்ஸ் ஒரு குளுக்கோஸ் மாலிகுல்ஸை கிரியேட் பண்ணனும், அவங்க எத்தனை கார்பன் மாலிகுல்ஸை ஃபிக்ஸ் பண்ணணும்? 6 கார்பன்ஸை ஃபிக்ஸ் பண்ணணும். 6 கார்பன்ஸை ஃபிக்ஸ் பண்ணனும் என்ன பண்ணணும்? You have to add that. You have to multiply into 6. So if multiply பண்ணீங்கன்னா that you will get 54 ATP out of it, because 3 ATP into 6 into 18 ATP into 3 is equal to 54 ATP, and here you can see 3 into 12 NADPH is equal to 36 NADPH. So 54 ATP 36 NADPH required. எதுக்கு? 3 molecules of glucose fix பண்றதுக்கு. 1 molecules fix பண்றதுக்கு எவ்வளவு தேவைப்படுது? 3 and 2 NADPH. But here the glucose structure is C6. That is important. C6H12, 6 carbon molecules. That's why we are adding 6 here. Is it clear, students? So answer is 54 ATP and 36 NADPH. Okay, the next question we are going to talk about: Rubisco act as a carboxylase and oxygenase, and respectively when concentration of CO2 and O2 is very low. Here this is the functional question. You have to be very clear. Functional question. Okay, so concentration of CO2 and O2 is very low, and concentration of CO2 is more and concentration of O2 is less. That is second option. O2 is more and CO2 is less. Here both will be more. Here see the question first. Rubisco, what is actually the Rubisco? You have to be very, I already told you that each question, each word is important, that you have to be very clear and you have to make sure that you know the words. Okay, and Rubisco is the most abundant enzyme in the world in organisms, especially in plants. And here you can see the Rubisco is actually what type of function they are doing. They are doing incorporation of CO2 molecules. I mean the carbon molecules mainly. Okay, and that incorporation of molecules will be conducted by the Rubisco enzyme. Rubisco enzyme. Enzyme means what? That only will enzyme which actually do conduct the reaction, catalyze the reaction, speed up the reaction. ஒரு yield product create ஆகிறதுக்கு, ஒரு result create ஆகிறதுக்கு, அந்த reaction-ஐ follow பண்ணி கொண்டு போகிறதுக்கு யூஸ் பண்ணுறது என்னது? enzyme. Here the Rubisco is a kind of an enzyme. என்னவா அது ஆக்ட் ஆகுதுன்றாங்க? கார்பாக்சிலேஸாவும் ஆக்ட் ஆகுது, ஆக்சிஜனேஸாவும் ஆக்ட் ஆகுதுன்றாங்க. Before you are going to understand this question, you must very clear with the word carboxylase-னா என்ன, oxygenase-னா என்ன? Carboxylase oxidase. Carboxylase oxidase is the kind of an enzyme which fixing the oxygen molecule, carbon molecule into the organic elements. Is it clear? So the chemical compound kulla the carboxylase oxidase enzyme oda work enna oxygen molecules fix pananum, carbon molecules fix pananum. So carboxylase oxidase both will be very important in photosynthesis. We are discussing about plants, physiological process, you have to imagine, and concentration of CO2 and O2 is very low when compared to that. That is the answer. In CO2, second option, third, fourth is the option. So what is exactly the Rubisco? Carboxylase act ஆகுது, oxygenase act ஆகுது. எப்போ concentration of CO2 and O2 will be more, ரெண்டு ஃபங்க்ஷன்ஸும் அதிகமாக இருக்கும்போது தான் O2 கான்சன்ட்ரேஷன் கார்பன் டை ஆக்சைடு கான்சன்ட்ரேஷன் ரெண்டுமே அதிகமாக இருக்கும்போது தான் ருபிஸ்கோ என்சைம் என்னவா ஆக்சைட் ஆகுது, கார்பாக்சிலேஸும் ஆக்ட் ஆகுது, ஆக்சிஜனேஸும் ஆக்ட் ஆகுது. யூ ஹேவ் டு இமேஜின் there is a function. What sort of a function is taking over? Here you can see photosynthesis takes place. What exactly photosynthesis takes place? Photosynthesis எதுக்கு நடக்குது? Carbon molecules, oxygen molecules will be get fixed. That is also a point of information. And carbon concentration of CO2, O2 is more. எப்போ concentration of CO2, O2 more இருக்கும்? When the stomatal regions are closed. You have to be very clear with information. Stomatal regions are closed. O2 வெளியில போக முடியாது, உள்ள கிரியேட் ஆகிருக்கிற CO2 என்ன பண்ண முடியாது, O2 உள்ள வராதனால வாட்டர் ஸ்ப்ளைசிங் ப்ராசஸ் நடக்காதனால அங்கே ப்ராசஸ் போகாது. ஸோ அப்போ என்ன பண்ணோம், Rubisco will go and act as a carboxylase and oxygenase and fixing the carbon and oxygen into the organic compounds. That is why the Rubisco is acting as a carboxylase as well as the oxygenase in concentration of O2 and CO2 is more when that is the correct option. Okay, and here the next question we're going to see, for the synthesis of one glucose molecule the Calvin cycle operates. How many times the Calvin cycle is operate? Calvin cycles is one of the most important cycle takeover in photosynthesis plants. From that only we are getting the carbon molecules. The carbon molecules will be utilized and converted into carbohydrates. So with the use of ATP and NADPH. So 2 times, 4 times, 6 times and 8 times. So 6 times. Why there is a 6 time is a correct answer? Just few minutes ago we discussed about one question. What is the question? How many number of molecules is required to create a 3 molecules of glucose from ATP and NADPH? So 6 atoms required to create 1 glucose molecules. 6 atoms. What is that? C6. So C6 molecules will be created, is required. So create what? Exo sugar molecules. The 6 carbons are the required agarapo. The Calvin has to cycle 6 times to create a 6 times of glucose molecules. That is very simple process you have to remember. So the synthesis of 1 glucose molecule, 1 glucose molecule. So the Calvin cycle operates with the 6 times better to create a glucose molecules information, which is actually exo sugar. So sugar molecules only the ultimate product of the photosynthesis process taking place in the plant system. That's very important, you have to remember. And now we're going to talk about the chlorophyll molecule at its carbon atom 3, the pyrrole ring 3 as one of the following. So இது என்ன actually பார்த்தீங்கன்னா இந்த questionல chlorophyll A is a kind of a pigment. You have to be very clear with that pigment. Okay, the pigment, which the structure of the pigment, it's carbon atom 3 of the pyrrole ring 2. One of the following and the group oda mingle ஆயிருக்கு. என்ன குரூப்னு கேக்குறாங்க, carboxyl group-ஆ, magnesium attached ஆயிருக்கா, இல்ல haldehyde and methyl group-ஆ. So first you have to be very clear with what is exactly the structure of chlorophyll and how, where is the carbon atoms or is the pyrrole ring 2. All these things will be important. Here you can see that the carbon C55H72O5NH4, the magnesium is the formula for the chlorophyll, which contains the chlorine ring in which the magnesium is surrounded by the 4 nitrogen atoms and which are attached to the several hydrocarbons. Hydrocarbons long chain. Hydrocarbons attached ஆகிறதுனால தான் அது என்ன ஆகுது, long chain create ஆகுது. And here you can see it helps to attach with the other hydrophobic protein. What is mean by hydrophobic protein? Hydrophobic, 2 different types of information are there. Hydrophilic substances, hydrophobic substances. Hydrophilic means what? It can easily attach with the water molecules, but hydrophobic is not easily attached. So that will be hydrophobic protein, the protein in thalamus region, where you can see that thalamus region of information. And the methyl group is only CH3 is getting attached to the pyrrole ring 2. Here you can see that structure, pyrrole ring 2 next to what? The chlorophyll A molecules at carbon atom 3. So here you have to be very clear with the structures information, chlorophyll A and what is exactly the structure, and chlorophyll and atoms, how many atoms are there? Atom 3, the carbon atom 3, which pyrrole ring of 2, and what is exactly for this is actually memory based question. Memory based question. Okay, so the memory based questions in the structures ninga chlorophyll A molecules or structures enna panano, you have to be remember. So that is the thing you have to easy for you to go for it. Okay, so next we're going to talk about a law of limiting factor was proposed by the Liebig, Achenslag, Arnon and Blackman. This is the example of history based question. So I hope that in this session we have included all the types of questions that what we have taught just few minutes ago when the session is on. So the Blackman is a kind of a person who proposed what? Law of limiting factors. I already told you that in the first question, what is exactly the factors? The factors is required for certain process to yield a product, to yield a result. That is very important. And Blackman's is 1905 enna pannirkaru, law of limiting factors are proposed pannirkaru. So he proposed that the number of separate factors, the rate of process is limited. Phase of the slowest process. See, the factors which present is minimum amount la irundaduna, law of limiting factors will inhibit the function of process and the yield also will get inhibit, the result also will get inhibit. So that is the law of limiting factors. In case of photosynthesis, what is exactly the factors? The light, okay, the leaf structure. The leaf structure required a proper leaf structure requirement. It should not get any pathogenic infections. It should not get any the water transportation is important for the leaf material. So leaf material water content irundada photosynthesis process நல்லபடியா நடக்கும். Photosynthesis process நல்லபடியா நடக்கணும்னா water is required on the leaf surface. So that is very important. And here the slowest process will be limited by the process, the rate process by the separate factors. The factors which are all inhibiting the product production, that is in the physiological process. So minimum number of amount in the process, in the factor minimum amount la irukko, adudha or process inhibit panno, that will be the law of limiting factors will be proposed by Blackman's. Okay, is it clear, students? Is it clear? So we come to the end of the session, and we have seen that 13 questions, and these 13 questions comprise the history and analytical, structural, and all those types of questions in photosynthesis topics we have seen. Okay, so the next session on coming Sunday we're going to see you, and I welcome you all to the session again. So the human physiology we are going to see. Human physiology, digestion and absorption we are going to see. That is on the topic lume exacta என்ன மாதிரியான questions எப்படி அந்த questions சால்வ் பண்றது, எப்படி அதோட structural functional technical oriented எப்படி நம்ம அதை அனலைஸ் பண்றதுன்றத we are going to see in the next session. Thank you so much for joining the session in the live YouTube la join பண்றதுக்கு ரொம்ப thanks. And next we're going to see, and 13 sessions இருக்குன்னு சொன்னாங்களா, I've told that the upcoming weeks oda timetable will be in the description. You can see that, and you can download the timetable, and you have to be very clear, and you have to join, and it will be very interest for you to if you join that particular if you have any doubts you can give me that, no problem. So we'll be joining, so we'll see you in the next session, okay, in the live session.